is associated with the DRGC-Ion channel group led by Michael McManus and Lily Jan at UCSF. Sorry for that.
That's okay. Thanks, Larry. Thanks to everyone for the invitation. I think transitioning from the last panel discussion to now, I made a lot of notes and I have a tendency to go off script. So if y'all catch me going off script, somebody just hold my hand and squeeze it. And I'll try to get back on the tracks. But I think the questions that we were talking about molecular probes and stuff are apropos to my talk. So this is the IDG ion channel gene list, 102 to begin with. And roughly a third of those are potassium channels. So the potassium channel alpha subunits, these are the pore forming subunits, are in purple, and the beta modulatory subunits are in green. So taken together, about a third of the IgG ion channel list is potassium channels, and probably one of the reasons is the potassium channels are the largest and most diverse family of ion channels. And my talk is really going to focus on what I refer to as putting the drug in the druggable genome, where the D in IDG, at least I think, means druggable. And I've taken this potassium channel list and whittled it down a little bit, and these are the ones that we're engaging on to begin with from the standpoint of trying to develop molecular probes. So the ones that are in blue are in the family referred to as the silent subunits. I'm going to talk about those a little bit more in detail. And then the ones in orange are part of a family of channels, sometimes referred to as the slow channels. So silent K is first. So silent K is a name that I don't like, yet I'm continuing to propagate it. And I think one of the reasons is that it's called silent K is we don't know much about how these things work, and thus they are part of the IDG list. So 7 of the silent K members, 7 of 10, are on the IDG list. They don't form channels on their own, so that's why they're called silent subunits. So they look like alpha or pore forming subunits, but when you express them by themselves, you don't get active channels. But if you express them with other voltage gated channels, most famously the KV2 channels, KV 2.1 and 2.2, you get channels that are voltage gated potassium channels, and their function, their things like the kinetics and things are different than the KV2s by themselves. So by forming these heteromeric channels, you can enable structural, functional, and pharmacological, and I've got a question mark there, pharmacological diversity. And that's one of the things that we're hoping to be able to do, is to generate selective pharmacological tools as part of a toolkit to help illuminate what these silent potassium channels do. So there really aren't any good specific pharmacological or genetic probes. And I think this is a great opportunity. These channels too are a great opportunity for us to learn from disease. So if you look at potassium channel disease, related potassium channel mutation related diseases are popping up more and more, not surprisingly, as we get more genomic sequences. And there's some themes that emerge in terms of the types of mutations that cause disease. A couple of the most kind of famous ones are ones that cause the channels not to fold properly, so you get hypofunction. Ones that cause the channels to have eroded selectivity, so now they don't show much of a preference for potassium and allow sodium to go through. So that essentially kind of perverts the function of the channel. And then ones that are actually gain of function mutations, where the channel is open at times that it normally wouldn't be. And I think by learning from disease, it gives us an opportunity to try to take some of the mutations that we know about in other potassium channels and move those into potassium channels that we know less about, and then bring those into animals, for instance, to see if we can provoke phenotypes that we wouldn't normally be able to provoke with things like knockouts. I'm not going to talk about that today other than what I've just done, but I think that's an important part of the toolkit. So this other family that I've called slick, slack, and slow, they're all members of a gene family. They're sometimes referred to as the slow genes, named after the first member of this family, which is a Drosophila mutation known as slowpoke. So the ones I'm going to focus on today are KCNT1, predominantly to a lesser degree KCNT2. So these slack channels can form homomeric channels, so can slick, and they can form channels, heteromeric channels by themselves. And then there's some speculation that these MAX-EK channels, which are formed by SLO1, might also be able to form heterotetrameric channels with some of these other family members. Today, though, I'm going to focus specifically on SLAC. And one of the reasons that I'm doing so is this is an interesting gene. So if you go back to when the IDG list was first written and you jump forward 3 years or so, a lot has happened in the life of the SLAC channel. So one of the most notable things is now we have over 30 gain-of-function mutations in KCNT1 that are associated with a number of severe pediatric epilepsies. So these are gain-of-function mutations. These channels are actually on more than they would normally be. And they're dominant gain-of-function mutations. So if you have one of these mutant SLAC channels, and with wild type, you still get a gain-of-function. But unfortunately, no specific pharmacological probes. Interestingly, though, because we know about these 30 mutations in humans, they essentially map to 3 different regions of the channel. And over the past few years, my group and a couple of other groups have made 3 different mutant mice. We kind of all went in our own directions and did this, as far as I know, without any coordination. And we actually chose mutations from different regions of the channels, these 3 kind of different modules that get mutated in human disease. And as it turns out, all of the animals have an epileptic phenotype. So that's really good from a standpoint of building a toolbox. So now we've got human mutations, we've made those mutations in animals, those animals have epilepsy phenotypes. If we can get some small molecule tools, then we can start asking some questions about whether or not there might be a productive mode of pharmacological intervention for the treatment of these epilepsies. So high throughput screening, in this case, we're talking about functional high throughput screening to try to find modulators of these channels. For potassium channels, our favorite mode is the thallium flux assay. So this is supposed to be a cell. This is a potassium channel. We can load the cell with a thallium sensitive fluorescent dye, and this is exactly the same concept of a calcium sensitive fluorescent dye like the flow and fura dyes. So we can load the cells up with these. We can put thallium on the outside of the cells, and thallium will flow down its concentration gradient through the channels and bind to the dye and get brighter. So in a scenario like this, the degree to which the fluorescence gets brighter is proportional to how many channels are open. So in this particular case, I've gotten this KV2 channel that I mentioned before. In this case, it's homomeric, and I've just exposed it to a fixed concentration of thallium and varying concentrations of extracellular potassium, because these are voltage gated channels. So as I depolarize the cell, I get more thallium flux. So you can see I get this nice graded change in fluorescence that's reflective of the activity of the channel. And so we can use kinetic imaging plate readers to make these assays high throughput. So in this case, we can take a plate reader that can acquire data from an entire 384 well plate at the same time, fairly high frame rates. And then the experiment's a pretty simple one. You put on the compound you want to test, you add thallium, and you sit back and watch what happens. And in this case, we're either looking for thallium flux to increase if we're thinking about an activator, or decrease if we're thinking about an inhibitor. So the pipeline is pretty simple. We're going to get, in the case of what we're talking about here, some compound libraries on the order of 60 to 100,000 compounds. And we're forwarding 4 molecular targets to begin with, or 4 ion channel targets to begin with. KV 2.1, as I said, it kind of is a necessary partner for the other silent subunits. So if we're thinking about developing a selective probe for the different silent subunits, one of the things that we need to understand is the pharmacology of KV2. And even though it is, in many cases, the dominant inward rectifying potassium channel in many, many mammalian cells responsible for action potential repolarization, there really aren't any selective tools for KV 2.1. So even though it's not an IDD channel, coming up with some probes for it, we would consider to be an important bonus. We put it in with KV 9.1. So of all of the different 10 KV, silent KVs, 7 of which are on the IDG list, we picked 9.1 primarily because it's the one where there at least is some amount of data suggesting that it may be important in either pain sensation or development of neuropathic pain syndromes. So it seemed like a reasonable place to start. as we're thinking about, building a toolkit of information. Then we chose SLAC for the obvious reasons that I told you about, and we chose the human mutation that's most prevalent in a form of epilepsy called malignant migrating partial seizures of infancy, MMPSI. So this is the one that's the most prevalent in association with this human disease. And the reason we did this is we wanted to make sure if we found any modulators of the wild type channel that they could also modulate the mutant channel, because it's not a foregone conclusion they would. In my experience, I've had plenty of instances where a single amino acid could make all the difference between whether an inhibitor worked on channel X versus channel Y. So we kind of thought that was a safeguard. And in fact, I'm not showing it here, but we did pilot screening on 2 other mutations from the different kind of mutational modules or cassettes that make up the places that are mutated in human disease. So we fed these things into the hopper together. We did all 4 channels. We did all 4 of the channels in parallel, and let's just say that we got some hits, and we've got some hits that look like they have some interesting selectivity profiles. I'm going to quickly talk about the ones that we've brought the furthest forward. This is one of the SLAC hits. This is an inhibitor. Okay, got it. This is an inhibitor with the idea, because they're gain of function mutations in the human epilepsies, that we would want an inhibitor to modulate that phenotype. This just shows that the thing that we found in thallium flux works in the gold standard voltage clamp electrophysiology. This is the gain of function mutation. This is the wild type SLAC channel, and this is both in the presence of our inhibitor. Then we took a step forward where we're looking at the calcium oscillations in cortical neurons. So if you have a dense population of cortical neurons in culture, they will spontaneously develop these synchronous calcium oscillations that can be further provoked into a more epileptic type phenotype with a variety of different means. In this case, we're lowering extracellular magnesium. And so this is baseline, and this is after we put on our test compounds. So after we put on our test compound, we can decrease the spike rate by about 50% in a concentration dependent manner. And even though I'm not showing this, this potency range is pretty much exactly what we see in electrophysiology and thallium flux. And as part of building a toolkit where we try to make a case that this phenotype that we're seeing is specific for SLAC inhibition, we take a molecule that's very closely related. Essentially, we flipped this amide. This one, by electrophysiology and thallium flux, we know is stone dead at SLAC, and it's also stone dead in this assay, so it doesn't do anything. That doesn't prove that our molecule is having its effect through SLAC modulation, but it begins to build that case. And we are building a library of a number of related molecules and of different chemotypes to start asking this question over and over again. Can we build a case that this effect that we're seeing in these neurons is SLAC related? So going forward with Silent K, we've got about 50 in total activators and inhibitors from a 60,000 compound screen. We're starting to engage medicinal chemistry to try to help us suss out which of these might be the most productive molecules to move forward with. An interesting thing that is kind of a side note, as I said, these silent subunits are referred to as silent subunits because they can't form alpha channels on their own. We're going to test that by actually exposing cells that are expressing just the silent subunits to our modulators, in this case, our activators, to see if anything happens. So why would we do something like that? Simply because in the past, I was dealing with a family of voltage-gated potassium channels, one of which wasn't active on its own, and we put some small molecules that were activators of members of that family on, and lo and behold, it was able to turn on the so-called silent subunit. So these might be really interesting probes in KV2 knockout animals. We're going to continue screening this silent subunit family. I already mentioned the possibility of genetic probes based on dominant, negative, and gain of function mutations. SLAC, we're moving forward with the activators that we found. The plot just shows that this one particular compound completely relieves the voltage dependence of the channel, just turns it on. It's not a voltage gated channel anymore. Moving toward in vivo probes and genetic probes, including pharmacoresistant animals. Thanks to Brittany Spitznagel and Kyle Emmett, who conducted the SLAC project, Silent K project, Sunny Dew, the IDG team, most importantly, Michael and Lily, and Olga Golyeva at UCSF. Olga made a lot of the cell lines, Vanderbilt and the Institute of Chemical Biology. And the SLAC work was also supported by an R21, R33 phased innovation